Welcome back everyone to the Immunisation Coalition's 2021 annual scientific meeting. My name is Margie Danchin and I'm going to be the facilitator for the fifth session of the conference. My background is I'm a paediatrician and clinician scientist at the Royal Children's Hospital, the Murdoch Children's Research Institute and the University of Melbourne, and I'm the leader of the vaccine uptake group. So during this session, we're going to have three presenters. We're actually going to have a question and answer session after our first speaker, and then another Q&A session after our second and third speakers. I'd like to introduce our first speaker, who is Professor Paul Effler. Paul is a public health physician and the Senior Medical Advisor for Immunisation in WA in the Department of Health. And he's also on the steering committee for the WHO Global Outbreak and Response Network. And he's going to be talking to us, very importantly, on the challenges facing distribution of COVID vaccines. So welcome, Paul.
Thanks for the opportunity to speak with the group today. It's obviously a critically important topic as we go forward in addressing the pandemic. So conflict of interest, I have no stock holdings, no pharmaceutical affiliations, and no pharmaceutical payments in my history. So when I think of the challenges in distributing the COVID vaccines, I really see that there's sort of four components of this. Some of it is inherent to the vaccine manufacturing processes, some of it to geopolitical issues, some of it to our public health priorities, and then also competing priorities as we go forward. So I'll address each of those briefly. And I did watch the session on vaccines last night, so I won't overlap with the vaccine explanations much, but I do think there's some important topics to bring up relevant to the distribution issues. So we have two vaccines in our immediate future in Australia, as we all know, the Pfizer BioNTech vaccine and then the Oxford vaccine, AstraZeneca, seem to be the ones that are, well, Pfizer's already approved and we're expecting a decision on AstraZeneca shortly. So turning first to the Pfizer vaccine, as we know, it's an mRNA vaccine, new technology, where essentially you introduce the mRNA into the cell, and then the cell makes the spike protein, the full spike protein after the mRNA is entered. And how it gets in there is with a lipid nanoparticle. And it turns out that these are really quite small, and this is technology I know Moderna, who is another mRNA vaccine manufacturer, they've been working on this technology for a long time for Nipah virus vaccines. But essentially you have a fat bubble which has some DNA in it, and when that lipid bubble reaches the cell, it can fuse with the cell membrane, deliver its payload of messenger RNA, and then that it goes inside. As it emerges, the messenger RNA goes in, hits the ribosomes, and then it ultimately gets turned into the spike proteins, which are then available for the immune system to attack. So what makes up that lipid nanoparticle? I can only pronounce two of these, and that's only if I use PEG as an abbreviation and cholesterol. But essentially, I bring this up to say that the lipid bubble is different between Pfizer and Moderna, so they're not exactly equivalent in the way that they can be handled and distributed. In fact, Pfizer is appears to be the more labile of the two, with the conditions required to maintain it after the vaccine has been made. So let's just touch briefly on the storage and handling. It needs to be kept at minus 70 degrees Celsius. We've all heard that. It can last up to 6 months in specialty freezers for that purpose. And here we see those specialty freezers in the manufacturing facility in Belgium, fairly impressive. And that worker there is holding what's called the pizza boxes, which I'll show you in a second, which is how the vaccine is packaged and distributed. Those pizza boxes cannot be broken down for transport, but only later on when they reach a destination point. It comes in vials, obviously, multi-dose vials, depending on your ability to withdraw material. You get 5 to 6 doses per vial. And those trays, those pizza box trays are 195 vials. So essentially you're getting almost 1,000 to maybe slightly less than 1,200 doses out of each tray. When you want to move it though from the factory, it's shipped in these thermal shippers that have been, specifically designed for this. And essentially, you'll see those 5 pizza boxes there. Number 2, these can hold up to 5 boxes. They don't need to hold 5 trays, but up to it. And then essentially, the other major component is dry ice, which is put in on top of this to keep it, at minus 70 degrees. These can be used to store the vaccine for some time if you keep topping up the dry ice, and they are reusable. But if they're filled with 5 trays of vaccine, they weigh 35 kilograms. So, something to be reckoned with as you move it around. And when they arrive on site, then obviously they need to go into minus 70 ultra low temperature freezers. And these will be purchased and distributed throughout Australia by the federal government. I know our state is purchasing some of its own just to have a little bit more flexibility in sites and how we might use it. And I imagine other jurisdictions may be doing the same. So once you get it to the site, you can thaw it out and keep it in a refrigerator, a regular vaccine refrigerator that's 2 to 8 degrees for 5 days. But that 5 days is good, but it's also a pretty short leash compared to what we're used to using in vaccinations. And once you add the diluent into it, you have 6 hours to use it, and really the sooner the better. So 5 days once you've thawed it, and then 6 hours once you've put the diluent in it. Again, pretty tight constraints compared to what we're used to. And so what is Australia expecting to get? It was 10 million doses up till last week, but now another 10 million has been secured. So theoretically, Australia could be receiving 20 million doses of Pfizer vaccine, all of that produced overseas. And as most of you will have seen this slide, this is the federal government's plan for the rollout. And just with the Pfizer vaccine, we're talking about, they're saying 30 to 50 hospital Pfizer vaccine hubs in urban and rural areas. And some of the jurisdictions, including my own, have started to publish what hospitals those will be. And those hubs will receive the vaccine, be able to keep it in freezers, and then ultimately be able to use that for in-reach vaccination clinics if they need to, and then use as a secondary distribution point for putting it out wider than the 6 specific hospital locations. So let's turn quickly to the Oxford Zeneca vaccine. As we all know, this is a non-replicating viral vector. And I sort of regret that they use vector, because I think that implies pathogenicity or disease state. But really, this is a chimp adenovirus that's been modified so it can't replicate in humans. And therefore, it really serves more as a Trojan horse. And this slide I just bring up so you can see the adenovirus coat over there. So the difference here is they've put the DNA for the spike protein into the viral vector, and it's much sturdier than that fat bubble that Pfizer's using. So this has some really wonderful characteristics as far as distribution and shipping. So again, the DNA goes in, the viral vector goes into the human, it merges with the cell, releases the DNA that ultimately goes on to make mRNA, and then ultimately that goes to make the spike protein. But the beauty of this vaccine is it's basically refrigerator temperature, refrigerator stable temperatures, 2 to 8, what we always expect, what we're experienced with, and it has a multi-dose vial that it's shipped in, but you can actually store that in a refrigerator and use it over 2 days. If you need to. So this really expands the opportunities to put vaccine out in places that are far removed from central hubs and minus degree 70 freezers. And, worldwide, a vaccine that is stable at regular vaccine temperatures could really make a huge difference to the pandemic. So what are we supposed to get of this? 3.8 million doses delivered from overseas initially, but another 30 million doses manufactured in Australia by CSL. And this will become important as we go forward and thinking about distribution, because we get onto the geopolitical issues about the COVID vaccines. And I'm sure you've heard this discussion going on, how rich countries are hoarding the COVID-19 vaccines Very few doses in the developing world, I should say, resource poor environments thus far. And you can see that photograph in France where when you're a wealthy country, you have the luxury of transporting them around by helicopter to remote areas if you need to. And with first dibs on vaccines, rich countries have cleared the shelves. The US, Britain, Canada, and others are hedging their bets and reserving huge numbers of the vaccines that are larger than their populations, while poor nations struggle. And if we look at that, as far as the proportion of vaccines secured or agreed to purchase by country as percent of population, we can see Canada has somewhere on the order, can do its population 4 to 5 times over, maybe even up to 6 times. The United States close to double the number of doses it has to population as far as fully vaccinating them. And you can also see Australia's there in the top 5, which we are securing enough vaccine to vaccinate everyone with the required number of doses twice over, theoretically. Now, some of that was done, of course, to hedge bets that some vaccines might not turn out to work or might have adverse event profiles that were unacceptable, but it really does leave a picture of the developed world, the rich countries, sucking up the vaccine. And in Australia's case, there are not as many, hardly any cases compared to other jurisdictions. It's a challenge, I think, as far as the optics of that. So where are the vaccines being used? Mostly, the largest number of doses in the United States, followed very closely by China. But you can see Europe has some, Germany, France has some vaccinations going on, but they've been constrained because they haven't been able to get all the doses that they want in the European Union. And so they've actually threatened to sue the vaccine manufacturers to make sure they keep up they get their doses as they were ordered. So this is a new development. And then the European Union is actually countered by saying we can introduce export controls so the vaccines that are manufactured in the EU countries can't be shipped out without approval. I think they've stepped back from that, but this really does represent a new wrinkle in the whole distribution planning, with countries suing and potential export blocks going on vaccines. And that's why the local manufacturing that CSL could do with AstraZeneca could turn out to be very important depending on how all this plays out. So this is a quote from Bruce Elward. It says, the worst possible outcome is you're offering vaccine to a whole country's population before we'd offered it to the highest risk persons in other countries. I believe Bruce is, I've known him for a number of years. I believe he's Canadian by background. But I think he makes a very important point that, we need to try and protect the most vulnerable in the world's community in general terms, or as a concept as public health professionals. So that has been thought of, and I'm happy to say there's a new initiative called COVAX, where countries can put money towards COVAX for an option to get vaccines and to support many different vaccine companies to develop products, and really with an effort to make these available to resource challenged countries so that they can get at least 20% of their populations vaccinated. So this is a relatively new effort. It's evolving every day from what I see as far as how firm the commitments are and when they might be releasing vaccine from COVAX. But, importantly, Australia is a partner in COVAX, and importantly, one of the wealthiest countries. The United States is not. But I hope this represents something for the global good, where we're able to get vaccines to all countries that need them, and just not have wealthy countries only benefit. Okay, and then I'll turn quickly to public health priorities. This also influences distribution more at a local level within a country. But essentially, in Australia, our initial priorities are for aged care and disability care residents, then the workers in those facilities, then the quarantine hotel and border workers, such at airports and ports, where there's a chance for spillover when people are returning from abroad, and then frontline healthcare workers, which would be expected to take care, diagnose and take care or test people that might have COVID. So when you look at this, you see that this isn't a situation where you probably are just going to put the vaccine out through normal channels, because you're really trying to target aged care and disability care. And that is going to be done by the Commonwealth in Australia with hiring up teams to do in-reach into those facilities. And again, quarantine workers and border workers, if you want to get high rates, which is the whole point of trying to ring fence exposure, then you're going to need to take the vaccine on site. Healthcare workers, a little bit more straightforward, but you need to, unlike flu, we're going to be dicing and slicing who gets first vaccines. So it says, just wanting those priority groups, that this is going to be managed by government initially in order to target those usually not our first cab off the rank for vaccination programs. So this changes though, you've probably seen it, this is the federal government's map of the different phases for vaccination. And what I was really talking about there was phase 1A, but if we go to phase 1B, we can see the number of doses is quite a bit more, almost 14 million doses for the country. And it expands to be persons 70 and above, and Aboriginal Torres Strait Islander people over 55 years of age or 55 and over, and other healthcare workers. So at this point in time, we really want to be able to distribute more broadly. And so that would largely be the AstraZeneca vaccine, which would hopefully, pending its approval, that it would go out through these select GP practices that are being identified by the Commonwealth through the public health, primary health alliances, in order to start vaccines in GP environments to reach this broader set of people. And finally, I'll just turn to competing priorities, and this will be very brief. What we have is the flu vaccination season is coming upon us, and as the number of doses seems to be of COVID vaccines coming into the country seems to be modest. Initially, it leaves us in a situation where the vast majority of COVID vaccinations might start to fall on a time when we typically are doing flu vaccinations. And, as you know, that's our big burst of vaccination activity every year. So overlaying these things, there will be a challenge. They are using different distribution networks if we're talking about the Pfizer vaccine initially, but ultimately you can see that you might want to be able to distribute the AstraZeneca vaccine, which is 2 to 8, through regular flu vaccination channels. And there's a lot of complications that could present with the timing of these, because you can't give them, it's not recommended that they be given at the same encounter, flu and COVID vaccines. In fact, there's a recommendation that there be a 14-day interval between them, if at all possible, though exceptions can be allowed. And so with that, I'll conclude my comments. And Margie, I'd be happy to take questions that we might have. Thank you.
Thanks so much, Paul. I think that's just raised so many issues. Thank you, that was brilliant. Firstly, just turning to some of the rollout challenges you mentioned around vaccine manufacturing, particularly for Pfizer vaccine, obviously the need for it to be stored at -70, and then it can be, in a 2 to 8 degree fridge for 5 days, but then only out for 6 hours. So, how do you think that's actually going to work on the ground for immunisers, particularly when you think of the urban-rural split? Is that going to be a challenge, and the number of providers that are going to have -70 freezers for the Pfizer vaccine?
Yeah, so what we're imagining in urban environments where it's a little more straightforward is that we could establish mass clinics, and basically we're calling them in-reach, you can call them outreach, where essentially the vaccine would be thawed at a Pfizer hub and then move that day to be used, hopefully that day, in a mass clinic. But we know we would have, up to 5 days to use that still at refrigerator temperatures. That's logistically easier to do when you have a capital that takes an hour to drive across or something like that. We can pretty much, ensure we can get it anywhere. I'll just speak about Perth, but pretty much similar to most of our capital cities. But when you want to get it out to, WA is really huge and has really remote regions, that's going to be a bigger challenge, and it's going to require, yet we will want, some priority group should get the first vaccine that comes out, and that's going to be Pfizer. So how do we get it to them? And I think that's going to take really careful planning about when we will likely ship thawed and then ship immediately with the planning already in place to use that vaccine when it arrives. Although I'm not ruling out we might ship at -70 right now. We're mostly thinking we're going to thaw and ship and use within 5 days and try and have that system really well oiled once we pull those vaccines out of the deep freeze.
So of course, there's all those challenges, and then there's the challenges around the use of the multi-dose vials, because that's not something that most Australian providers or immunisers are used to using on a regular basis. And Brendan Murphy certainly highlighted that, the issue around wastage very clearly in the media over the weekend. What are your thoughts around wastage and the multi-dose vials and making sure that, Brendan Murphy was saying that some hubs may potentially even be shut down if there was a lot of wastage. What are your thoughts around that and the actual use of the multi-dose vials?
Well, the multi-dose vials do present a challenge, but we did use them in 2009 for the pandemic, for the influenza pandemic. So it's not, and providers have some experience with other medicines in multi-dose vials. From an infection control standpoint, and I'm not an expert on this, but I think that it's minimal risk because you're basically drawing up the vaccines and then taking it to a station for immunization. So there shouldn't be a situation where you're going back to the vial after there's been contact with the patient. So I think that's pretty sorted, but it's how to get out the number of doses. And, initially it was considered to be 5, and now they've sort of raised the bar to 6, which makes drawing up those doses very important in trying to get low dead volume syringes and needles, which I understand are very short supply around the world. So from my standpoint, the most important thing to do is do it safely, right? And if we get 5 doses, great. And I think the biggest wastage is more likely to come through handling errors of boxes or whole vials rather than the dose not being extracted perfectly at the scene. That would be my guess.
Yeah. And of course, we're aware that all immunisers have to do the course, the training course with the 6 modules, which includes, the use of multi-dose vials and administration. That's the course directed or overseen by APNA. But there's also a lot of other issues for providers, aren't there? Particularly around now mandatory uploading of the vaccines to the air, and assessment of personal eligibility at the time of vaccination. How do you see those logistical issues being handled on the ground?
Well, they're 2 separate issues, sort of.
Yeah,
as far as uploading into air, yes. I mean, I do think that's imperative, right? We're lucky enough in this country to have a registry that covers everyone for vaccinations. It's a really important tool, and making sure that we know that they were vaccinated. We can then use that information to make sure the vaccines are safe by looking against different hospital data sets and emergency departments. We can also see, is the vaccine performing the way we expect it to? Down the road, it might give us an indication of how long that protection lasts, or if mixed schedules are better, or adding a different formulation of a vaccine is important for protection. So I think getting it into the register, I think that's something we've all been striving for. Now they've just said it's mandatory, but we should focus on the electronic systems to make them sort out. Yeah. And then your second question?
Well, so the second question was really around assessing personal eligibility. It's going to be tricky in some of these hubs with long queues. And I guess the answer would probably be for those people to go and see their GP or their healthcare provider for a slightly longer consultation. But just your thoughts around that, if people have questions at the time of vaccination about their own personal medical risk status?
Well, the own questions about their personal risk, I think that has to be handled individually, right? With talking to a knowledgeable consultant. But as far as a screening for people, I don't think that'll be too difficult. And one thing I would caution, and this may be out of turn, but we who work in the vaccination space and really believe in vaccine science think everybody's going to want these. Right. But I think we may find that there's a challenge. You'll reach a certain proportion of the population that's like, yes, give me that vaccine. But then we're going to have folks that are less inclined to get vaccinated. So with the numbers, I think we may have temporary controls here as when the numbers of doses aren't great coming in. But once that opens up, I'm thinking maybe the rigidity for which we screen people could also be loosened as a vaccine becomes available.
Yeah, that's a really good point. So John Litt has asked, as a guiding principle, shouldn't the highest risk groups all get the more effective vaccines, the Pfizer vaccine, the mRNA, the Pfizer or the Moderna vaccines? I think there's a real lack of understanding out there that people aren't really going to get a choice. Generally in 1A, they'll be offered most likely Pfizer, and in 1B, they'll most likely be offered AstraZeneca. But it's not like people are going to get to the vaccination hub and be able to choose which one they would prefer.
Yeah, absolutely. So we will intentionally try not to mix vaccines in any given location because you're going to get errors that way as far as the schedules with repeat doses. And I think, the priority population should get the first vaccine. It can be safely delivered in a potent format to their location. And, many times that will be Pfizer. There may be occasions because of the shipping and other requirements that would be AstraZeneca. And I think as a community, we need to get our heads around that the figures that have been produced in the clinical trials, while extremely encouraging for all the vaccines and for some amazing results, they may not necessarily be the exact figures we see when these vaccines are rolled out in real life. Because of variants and because of other factors that make real life different than clinical trials. So I don't want to, think it's helpful to get too hung up on the efficacy from these limited clinical trials. And we need to learn more around the world from real world experiences before we make those distinctions, if we need to make those distinctions.
Yeah, no, I totally agree. And just, I guess, leading on from that, just really touching on that fantastic point you made around the inequity and the requirement for the COVAX facility to operate effectively. There's obviously a moral imperative that we all have to make sure that the poorer countries have equal access to these vaccines. But I've also heard it discussed that potentially if there's increased viral transmission in these poorer countries, there's a higher risk of vaccine escape and mutant viruses. So not only is there the moral imperative, but also we need to be getting these vaccines across, richer and poorer countries to try and reduce potentially the number of, more virulent strains. I'd be interested in your thoughts around the inequity issue, which is pretty galling.
Yeah, it is. It is galling, and in health, it's been that way for a long time. But I think a pandemic really brings it to the fore because it's acute and sharp, and you can see it. So I think the COVAX initiative is a fantastic idea. I do think we need to try and protect the most vulnerable. I may be speaking, I'll just speak as an individual, not a representative of a government. I think there may be a time when we need to face reality that these vaccines are good at hopefully preventing serious illness and death, but may not produce sterilizing immunity, and transmission may still occur, and ultimately the virus will evolve. But hopefully with a combination of vaccines and people that are younger folks that have had infection and survived, hopefully that will limit the amount of transmission ultimately so that we can manage it in our health system, have less people dying, but also limit opportunities for changes in the virus as it replicates.
I've seen it said that, richer countries should be aiming to vaccinate their highest risk groups, our phase 1A and 1B, the sort of top 15 to 25%, if you like, and then redeploy the vaccines that they've purchased to poorer countries. I wonder if that is really pie in the sky thinking, or whether something like that could actually happen.
It's a very noble thought, and I hope something close to that happens, although I guess I'm not holding my breath. There may come a time though where, I read an article in Science recently where they were talking about how the best strategy might be vaccinating the vulnerable, but letting younger people actually who don't get very sick get immunity naturally if you want to control transmission in the longer term. Yeah,
absolutely. And did you have any other comments that you wanted to make around the 1A and the 1B groups? Someone made a comment that GPs are not in the 1A group unless they work in a respiratory clinic. They mostly sit in the 1B group. So there is, I guess, a lot of confusion still, isn't there, about which groups people sit in? And that's going to be
so
quite
1A is pretty narrow, the tip of the spear. It's just those groups I talked about. So you're talking about a small number of doses in close hold, and it looks like it's going to be Pfizer. So it's even more important to have that well managed. But as we get to 1B, if we start getting a lot of a vaccine that's stable at 2 to 8 degrees, I really think there's an argument for really ramping up primary care's involvement beyond the selected GP practices in that initial wave that the Commonwealth is doing. That's a good way to start. But if we really, if CSL's production comes online and the AstraZeneca is approved, I can't see why we wouldn't use the amazing distribution system we have for all other vaccines that go to 2 to 8 and have the ability to deliver a flu vaccination program that's pretty phenomenal every year.
Yeah, absolutely. And just touching on the flu issue, Christoph Feil has asked, there is a risk that Australians will be confused about which vaccine to receive first, COVID or flu. How should government, industry, immunisation coalition clarify this issue for them?
Well, that's a very good and very difficult question. I think early on, we thought the COVID vaccines were going to come early enough where we would basically do that first because it is a huge priority, and then do flu vaccinations. Now that it looks like there's going to be significant overlap, I think we may be in a situation where as flu vaccines become available, we should start using them through the regular GP primary healthcare distribution channels as we await further word on the volumes of COVID vaccine that are coming down the pike to us. So It's a very difficult, pragmatic question. I'm hoping for some national guidance because any way you cut that answer, people are going to be unhappy. And it would be nice to have it really considered at a federal level to give us, those that work at the coalface, some real strong guidance to go with.
Yeah, and potentially more challenging. Paul Griffin raised the issue last night that ideally you'd separate COVID and flu vaccination by about a 2-week period for people so that, if there were adverse events, there wasn't that confusion. It is going to be very challenging on so many levels, isn't it, to get these vaccines out?
Yeah, I've sort of intermittently been trolling, trying to find maybe data from the US where they had flu season and COVID vaccines to see if people were actually giving them simultaneously or studying that, but I haven't really seen anything yet. It would be wonderful if we could and they worked, but I think at least for the next few months, we'll be talking about keeping them separate, as you said.
Yeah, fantastic. All right, well, thank you so much, Paul. That was just brilliant. Of course, we haven't got through all of the questions. There were many more for you, but we have to leave it there. So thank you so much for your time today.
Thanks for having me. Really appreciate it.
See, Paul. Thanks. All right, so I'm now going to introduce the first of our next 2 speakers to everybody. And he doesn't really need much introduction. So Professor Lou Irving. As you know, Lou is a respiratory physician at Peter Mac Cancer Centre and the Director of Respiratory and Sleep Medicine. And he's going to talk to us today about healthcare worker infections, which is a critical topic. So thanks so much, Lou.
Margie, thanks. And I'm going to describe our experience and lessons learned. And before doing so, I just want to acknowledge the traditional custodians of the land that we're speaking on and pay my respect to elders past, present and emerging. I also want to acknowledge that there may be people, healthcare workers listening who have actually had COVID, and I hope this talk isn't distressing or opens any wounds at all. I'm going to briefly talk about an outbreak of healthcare worker infections at the healthcare service that I work at. I'm going to briefly talk about managing post-COVID symptoms in those healthcare workers and measures that are being taken to try and reduce the risk to healthcare workers. We know from overseas report that healthcare workers are at risk of infections, and a number of causes have been postulated, including unavailability or inadequate PPE, exposure to large numbers of COVID patients, worker fatigue, and limited testing leading to healthcare worker contact with undiagnosed cases and not wearing appropriate PPE. The health service that I work at, Royal Melbourne Hospital, has a city campus and the Royal Park campus, the 2 areas that I'll be discussing. And there are about 10,000 staff. The first wave that we experienced was more a ripple than a wave. It occurred last March. It was a time when we had, in the order of 8 to 10 positive patients in the hospital at a time. And at the same time, we established a staff testing clinic and encouraged staff to present as soon as they had symptoms. And in a 4-week period during the first wave, there were 11 positive staff with symptoms. 8 had a clear history of travel or recent contact with a traveller. 3, there wasn't such a history, but in those, there was really no definite patient contact. And so we really concluded from this experience that during the first wave, community acquisition was occurring in healthcare workers rather than workplace infection. In contrast, we were hit by a second wave in July to October at a time when there was a significant amount of COVID in Melbourne, but particularly in our catchment area, and we had a significant number of COVID positive patients in the hospital. And during this time, there were 262 healthcare worker infections, predominantly in nurses, but also in support staff and doctors. And luckily, the large majority of illnesses were mild, but 15 of our staff were admitted, including 2 into ICU, and one young nurse required intubation for a number of days. Luckily, there were no deaths. There were several outbreaks, but the most telling came from the Royal Park campus, which is a combination of a geriatric and rehabilitation wards. And this outbreak occurred quickly after several COVID positive elderly patients from residential care were admitted. Appropriate infection control measures were taken, but they were being managed in open wards. And then within 2 weeks, there was a rapid increase in the number of inpatient infections amongst patients. And unfortunately, there was a 30% mortality in that group. And at the same time, an explosive number of healthcare worker infections, 40% of our total during the second wave. There were less dramatic outbreaks of COVID in our city campus, including on our hot wards with known cases, but also on our cold wards. The marked number of healthcare worker infections, particularly at the Royal Park campus, caused us to think about, what was occurring and why this seemed to be defying what was appropriate PPE at the time. And some of the conclusions we came to was that there were a large number of COVID positive patients being managed in open wards, and these patients that were potentially generating a large viral load. The nature of the patients in that they tended to be confused and they wandered, and they required a lot of nursing care compared to some of the other COVID patients, COVID positive patients at the city campus. We identified that some of these confused patients were calling out and coughing and crying, and this concept of aerosol generating behaviours we thought was possibly important. We wondered about a breakdown in infection control in doffing stations and in tea rooms, and we took measures to try and strengthen this. We were certainly conscious of staff fatigue because a large number of staff were being furloughed at this point in time. We were conscious that because of the reduction in staff, there were staff then having to fill spots across multiple sites. It was very difficult to maintain a constant workforce within wards. A couple of our staff, in fact, probably acquired the infection by living with colleagues and fellow healthcare workers. And we wondered about whether there were unexpected effects of ventilation and airflow in clinical areas. So these were sort of our thoughts, having seen this explosive increase in healthcare worker infections. And a number of actions were taken. Wards were closed, patients were transferred. We sort of mandated the use of single rooms. I should highlight that both campuses, the Rural Park campus and the City campus, had a very limited number of single rooms and only a small number of negative pressure rooms. We upgraded the PPE requirements above the DHHS guidelines. We inserted spotters at donning and doffing stations to strengthen the use of PPE. There was extra cleaning. There was rapid testing of symptomatic staff, and staff were furloughed until a negative test. We obviously supported the staff, and we took measures to investigate airflow and to clean air, and I'll come back to that. So not only was this a difficult time for us, but it also meant that we had a significant number of staff who required follow-up. And we established a post-COVID clinic, and it was for two groups of people, not just healthcare workers, but other patients. One group were patients who'd been admitted with significant COVID and who usually had evidence of resolving inflammation and fibrosis. And there's a CT on the right there of a patient who required follow-up, as any patient would with resolving ARDS. But then there was a bigger group of people who had persisting or new symptoms following what was initially relatively mild acute COVID. And those symptoms, consisted of, in general, of fatigue, exercise intolerance, sleep disturbance, palpitations, what's sometimes called brain fog, mood disturbance, grief, frustration, anger, and difficulty returning to work. So this wasn't just our own healthcare workers, but also members of the public who were referred to us. And in thinking about how to set up a clinic for this group of patients, we were informed by the literature on post-viral symptoms. And there's a large literature on possible mechanisms for post-viral symptoms, including biological, psychological, and social. And then there are sort of previous syndromes, including encephalitis lethargica, that followed the influenza pandemic of 1918, and that still isn't fully understood. And so we approached the clinic, this group of patients, with a very open mind, and we decided from the onset to have a multidisciplinary team for this clinic. And the structure of the clinic was that 3 or 4 physicians would see individual patients face-to-face or by telehealth, and it would be the same physician who would follow the patient over multiple reviews, and that the physician would focus on the symptoms that were important to the patient. And that at the end of the clinic, we would then have a multidisciplinary discussion of each patient. And in the construct of their physical, psychological, and social factors influencing that patient, and to try and understand which of those factors were influencing the symptoms. We used a functional test to get an idea of physical and psychological well-being, and we mostly used a 6-minute walk test, particularly when patients complained of exercise intolerance or fatigue, or palpitations with exercise. We sometimes use sleep studies in people who were concerned about a marked sleep disturbance, and sometimes a screening questionnaire of mood. And we followed up these screening tests with targeted investigations to rule out physical illness and to reassure the patient. We've been very careful to avoid diagnostic labels as much as possible. And in the healing phase, we've been using exercise physiologists to supervise graded activity and psychologists and GPs to support mental well-being. And our experience has been that overall, the prognosis for these patients has been very positive. Virtually all have recovered within 3 months, although recovery has not always been linear, and obstacles do occur. And if obstacles do occur, often there is a history prior to COVID of similar events. We don't appear to have been seeing a sort of the severe disease that has been reported from the UK and Europe. We've had other responses to try and limit healthcare worker infections, and we've formed a sort of a research and implementation alliance with the University of Melbourne, the Doherty Centre, CSIRO, and Western Hospital, and we've been looking at airflow in clinical areas using CO2 monitors and smoke. And it's clear that air doesn't always flow in the direction that you expect it to flow, and that, I think, is going to inform how we move forward. We've also introduced air scrubbers or portable HEPA filtration in areas where there are people with SCOVID or COVID, and the pictures there are part of our screening clinic, and you can see an air scrubber between the nurse and the patient, and also in the waiting room of the screening clinic. And we've been exploring the use of the McMonty Hood, and for those who aren't aware of the McMonty Hood, it's this device. It looks like a pram hood. It's portable. It sits over a patient, although there isn't a tight fit. And at the back of the hood is an extraction fan, very similar to what is in your bathroom to extract water vapour and steam during a shower. And the extraction fan then blows the air through a HEPA filter. And there's some data on the right showing that the air can be cleaned to a high level. The hood is the idea of Forbes McGain, who's an ICU doctor at the Western, and Jason Monty, who's head of engineering at Melbourne University. And we're just beginning to explore the use of these hoods at Royal Melbourne, but they have been extensively used at the Western. And I think, with portable air scrubbers, there is a role for devices like this, particularly if there is a lack of single rooms, if there's a lack of negative pressure rooms, in patients where you might want to use aerosol generating procedures such as BiPAP, CPAP, high flow oxygen and nebulization. So that's our experience with healthcare worker infections and the response to ongoing symptoms, and sort of an overview of measures that we're trying to take to reduce risk in healthcare workers. Thank you.
Thanks, Lou. Sorry about that little delay. So we might carry over the questions for your talk to the end. And we'll just have our last speaker for this session, who is Associate Professor Deborah Williamson. Deborah is a clinical microbiologist and Deputy Director of the Microbiological Diagnostic Unit Public Health Laboratory and Lab Head. And Deb's going to talk to us about diagnostic tests. Thanks so much.
Thanks, Margie. Thanks very much to the organisers for inviting me to talk today. And like Lou, I'd just like to acknowledge the traditional custodians of the land, which we meet today, and pay respects to elders past, present and emerging. And as Margie said, I'm going to be talking about diagnostic testing for COVID-19, and clearly this has been critical to the public health response. And this is really a whistle-stop tour, I guess, through the evolution of diagnostic testing for COVID-19, and it's for pathologists. It's been a head-spinning journey, I think it's fair to say, and really we've faced massive challenges actually in the laboratory. I guess spanning areas such as, who to test, how to test them, how to get the specimens to the lab in a tiny way when they get to the lab, which testing platform to use. We've faced supply chain issues around swabs, reagents, hardware. We've had to massively upskill a workforce to do this molecular testing on a scale which has never really been seen before, and maintain workforce capability in the face of really quite marked workforce fatigue. We've had to navigate quite complex regulatory frameworks. We've had to work out how to introduce new tests into testing algorithms, how to report results as quickly as possible to the right person. And also, as many of you will be well aware, and this has hit the front pages a number of times, how to interpret results, particularly the issues of false positive results, false negative results, how long are people infectious, what does it mean if you pick up a little bit of RNA, 14 days after somebody's had symptoms. So it's been, there have been very complex challenges and issues to work through in the laboratory. And, these are scientists from, some of the scientists, I should say, from my lab at the Royal Melbourne. Clearly the panel on the left was taken before any physical distancing measures. That was taken at the start of 2020. I'm glad to say the same people are still working. They're not looking as shiny and happy, I would say, but these laboratory scientists, they don't often get mentioned in some of the headlines, but these guys have worked around the clock in labs across the country and indeed across the globe. And I think of them as the sort of workhorse, if you like, the engine room of the pandemic, because put very simply, without a test, there's very little public health response that you can have. You need a result to have actionable public health responses. And so, these guys have done a quite outstanding job. One of the other challenges that we've had to navigate in pathology land has been, again, these constant new great tests that have, I guess, come onto the market, or actually not so much on the market in Australia, but more in the headlines. Myself and several colleagues, I think we touched base regularly, and our inboxes are inundated with requests to evaluate the next great test. And that has been a challenge because it's a fine line between I guess rubbishing a test, but also making sure that we are evaluating every option that we have. And there's been, certainly in my career, I cannot recall a time where pathology and laboratory testing has been as much in the headlines as it has over the past year. All right, so I'm just going to whiz through the main tests that we have used and continue to use for COVID-19. And clearly the one that is the gold standard and has served us very well and continued to serve us very well is reverse transcription PCR, so the gold standard molecular test. And these were first introduced in Australia and globally very early in January 2020. And then were rolled out really across diagnostic labs late January and throughout February 2020. One key thing that we have learned in the lab is to have a diversity of testing platforms. So we can't just rely on one machine or one supply chain to maintain a continuity of testing. We absolutely need a range of testing platforms because, if one goes down, then we can rapidly switch to another. And in the lab that I work at Royal Melbourne, we went from having one machine, one testing platform in February 2020, and now we're up to at least 8 different testing platforms. And we've learned the hard way that you need that range of testing platforms. A test that's received a lot of headline over the past year is the Rapid Gene Expert test. And these have been really incredibly useful, particularly in hospital settings and outbreak settings where you need a result within a matter of hours. And, we can provide a result back within 2 hours on a patient or on a healthcare worker. We were involved in the response that Lou mentioned earlier, and these have been fantastically useful tests. However, out of all of the tests that we use, this one has really suffered the most, I think, from supply chain issues, and that's because they are so useful and because the manufacturing is offshore. One thing that we tried to look at to try and mitigate these shortages, and I'll say a lot of the stuff that I'm going to talk about has been born out of necessity. So out of necessity, we've been forced to look at alternative testing strategies. And one thing that we looked at was whether we could pool samples and use them on this testing platform. So this is sample pooling has been an approach to upscale the number of tests. And so we looked at pools of 4 and we looked at pools of 6. And this is where you take samples from more than one patient, you put them together and you run the test, and you accept a slight degree of loss in sensitivity at the expense of being able to do more tests. And actually, we were quite pleasantly surprised by how this worked on the Gene Expert. And this is a protocol that we were able to share with some resource limited settings where clearly the supply issues are far worse than we have. Another novel diagnostic approach that we looked at Royal Melbourne way back in March of last year was the use of saliva as a diagnostic specimen. And again, this has had considerable press over the past year. Again, this was something that was born out of necessity. So early March 2020, different time, different context, quite different place. And at that point, we thought that we might run out of swabs to test patients. And we decided to look at using saliva as a diagnostic sample. The other reason that we wanted to use saliva was because there were emerging reports from the UK and the US of healthcare worker infections, and that was well before that second wave in Victoria that Lou described. And so we thought, well, if we can validate this as a diagnostic specimen, then it might reduce the risk to our frontline healthcare workers. And again, we compared that to the gold standard of a nasopharyngeal swab. And we found that, yes, there was a moderate decrease in sensitivity when saliva was used, but actually it was not it was not too much. And we thought, well, this is useful that we could have this in our back pocket. This was really the sort of alpha version of it. There have been modifications to this done by other groups since this work. Interestingly, you know, the saliva work has really snowballed. And there was a rollout, a small scale pilot rollout by the state government in June of last year in a couple of suburbs. But now this has really taken off, particularly in the hotel quarantine program, where hotel quarantine workers have daily saliva tests. And again, it's been rolled out actually across the country now, and to a range of other settings where frequent testing is needed. And, you know, clearly not everybody would be comfortable with a daily nasal swab. And we also looked at whether saliva was stable in a range of different transport media, and actually just in neat saliva at different temperatures and over different time periods. And again, we were quite pleasantly surprised to see that the virus could still be detected in neat saliva at room temperature up to a week following collection. Now, we wouldn't advocate that, but again, this was something that we were able to share with resource limited settings where transport and logistics, again, are really much more of a challenge than they are here. Another thing that we looked at was the use of 3D printed swabs as an alternative to, I guess, conventional swabs. And again, this was work that was done back in March and April of last year, born out of necessity, where we thought we might face swab shortages, and we wanted to have a sovereign supply chain of swabs. And we worked very closely with a 3D printing company in Melbourne. And 3D printing is not new in medical technology, right? It's used in medical devices and implants and molds. And this company actually came to us and said, we think that we can help here. And we've got a surge production capacity of up to 8,000 swabs a day. And we said, thank you very much. We're very happy to work with you and look at this. And one of the good things is that the material that's used in swabs, it's a material called nylon 2, and it's amenable to autoclaving. And so again, you know, we were at the epicenter, I think, at Royal Melbourne in many ways last year, mainly because of the second wave. But, you know, I think we did a lot of work where we looked at these innovative testing methods, and we were able to rapidly set up, I have to say, with Lou and others' help, through our screening clinic, a clinical trial to look at these swabs. This is just the prototype swab that we used. And, you know, the great thing about 3D printing is that you can modify things very quickly if you don't like it. And so, you know, we worked through a number of iterations of the flexibility of the swab, the break-off point, and we wanted to get it as close as possible to a flocked swab. And this was a prototype that we went with. And I won't walk you through this in detail, but suffice to say, they were no worse than the conventional swabs, and they picked up a lot of cellular material as well. And actually, these are now being rolled out across the state. I'll just talk very briefly about serological tests for COVID. Now, this is, I guess, not detection of the actual virus, but the antibody response to the virus. And the development of serological tests for COVID-19, I think it's fair to say, lagged behind the development of molecular testing. And that's really for a number of reasons. One of them being, it's never really found a home in clinical diagnostics. I guess the role of it was and still is relatively unclear in large-scale diagnostics for COVID. But also there were issues around cross-reactivity, mainly with other human coronaviruses. And so, you know, it lagged a bit behind the introduction into diagnostic labs. There are a range of different serological tests, all the way through from the gold standard of a neutralization test. And you'll have heard about that in a number of other presentations related to vaccine testing. So that is the gold standard test, highly specific, but can only be done in very specialized reference labs. And serological tests span from that all the way through to point of care tests. So these lateral flow finger prick devices, again, hit headlines in a big way last year. And so these devices essentially are, they look a bit like a pregnancy test. They're cartridge based, and you put some serum or plasma in the well, and it moves across there, and you get a positive, negative, or equivocal result. And again, these were rolled out on a relatively large scale, actually, in the UK, and a number of peak bodies in Australia really came out and said these don't really have a place in Australia. And our group at Doherty were asked to evaluate these tests by the TGA, the Therapeutic Goods Administration. We're asked to undertake something called a post-market validation study, which was basically a study to assess the real-world performance of the tests and compare them to what the manufacturer said was the performance of the test. We published on this last year, and again, in a nutshell, what we found, this is the results from the first 5 of those tests. We've done nearly 20 of these kits now, but when you looked at all time points following COVID, and these are looking at serum samples from around 100 positive COVID-19 patients. When you look at all time points, the sensitivities are actually pretty rubbish, but that's not surprising because it takes time to mount an antibody response. When you look at the sensitivity 14 days or more following symptom onset, the sensitivity rises generally for most of them around 90%. The specificity is not too bad, generally sits above 94%. And this work, as I said, is ongoing, and the results of that can continue to be found on the TGA website. I'll now just briefly cover rapid antigen tests, and these I would say are the latest tests which have received a lot of press. And again, these are cartridge-based point-of-care tests which are designed to detect viral protein from clinical samples. So they're not a molecular test, they're not an antibody test, but they look for the presence of the viral protein, generally in nasal swabs. So a direct detection of the virus. And they have a number of advantages. They're portable, they're scalable, they're quick, they take 15 minutes, and they're pretty cheap. They're $10 to $15 a test. But they are less sensitive than PCR, and that's because PCR has an amplification step to amplify up the viral nucleic acid, but this is looking directly for the virus. So you need quite a lot there to be able to detect the virus. And again, you know, I think it's fair to say that these, they capture the imagination as something that is conceptually attractive, right, to enable us to get back to some semblance of a normal society, in that, you know, you could test, and if you're negative, then you're okay. But that's not the case, because we know that the limited sensitivity means that we do get false negative results. And again, we were asked to look at the performance, the real world performance of these tests. And I should just say that these tests are designed to be used generally in the first few days of symptom onset, and they're most useful in high disease prevalence settings. And that's not here. You know, even during the second wave of COVID in Victoria, we were not a particularly high prevalence setting compared to many, many other countries. And so we had to look at the performance of these tests in a low disease prevalence setting. And we undertook a pilot study. We worked with, this is work that involved a huge amount of people late last year at Royal Melbourne, Monash and Austin. And we looked at people coming through the screening clinic and compared the performance of antigen tests to the standard of care. This was really challenging because it was at a time of almost zero community transmission, but we worked with the Victorian government to, I guess, access patients who were known to have COVID to gather some information on sensitivity. That study's in press at the moment. And essentially what we found was that the tests were around 90% sensitive in the first few days of symptom onset, consistent with what the manufacturer said and the intended purpose. And they were very specific. Again, what work needs to be done to look at the performance of these tests in a way that I think that people think about using them in a low prevalence setting, which is for large scale asymptomatic screening. And that's not how these tests are designed to be used here. And I'll finish by just talking about some work again that was done late last year at the University of Melbourne. This is work that was led by Tim Stenier at Doherty and a group of us. So this is moving testing out of the lab. Now, for a pathologist, this is way out of my comfort zone, but I think it's fair to say that most of us have been pushed way out of our comfort zone last year, looking at these different testing methods. And so this was using a pop-up laboratory, which was essentially a 20-feet shipping container fitted out to PC2 laboratory specifications. So it's got a biosafety cabinet here, it's got rapid molecular tests in here, it's got waste disposal, you can immediately see, again, the conceptual attractiveness of this in rural or remote or resource limited settings where you can essentially fit out one of these shipping containers to have a pop-up laboratory, which means that staff can conduct the testing in a safe environment with quality assured results, which are transmitted back to a central lab. And so this ran, as I say, at the university, on the University of Melbourne campus. Really, this was a proof of concept to look at whether decentralized testing was possible. And I have to say it worked extremely well, and we're now looking at a number of other sites for this. That's not to replace centralized laboratories, but again, it's to complement that. So what did we learn in the lab last year regarding COVID-19 testing? And I hope I've made the point that necessity really was the mother of invention last year. While PCR testing in large labs continues to be the mainstay of testing for COVID-19, we really had to look at different testing approaches. We did need a lot of redundancy and diversity of our testing platforms. We found that we actually needed to keep a store of high quality clinical material so that we could rapidly validate a lot of these different testing approaches, and that's important. So essentially, we didn't throw stuff away. We didn't throw positive samples away. We kept them so that we could evaluate new tests. We had to be clinically engaged and responsive, and that was absolutely critical last year. So we had regular meetings with our clinicians, with our public health colleagues, and it's not infrequent that on a weekly basis, testing algorithms had to change. We had to change the tempo and of testing, the type of testing, and you can only do that if you know what's going on the shop floor. And we learned the hard way that if you have downtime, it's not downtime, it's planning time for the next outbreak, for the next wave. And as I said, this work was really done on behalf of presenting this on behalf of a huge number of colleagues across a range of institutes. And thanks, Margie. I'm happy to take questions.
Thanks so much, Deborah. There's some really exciting innovation coming down the pipeline. I know I've got lots of questions, but it might go to you first, Lou. I just wanted to ask, of the 262 healthcare worker infections, there were nearly 70% in nurses, 68%, and only 8% in doctors. Can you talk about that a little bit? I mean, obviously nurses are more directly involved in patient care, but that seems to be a huge discrepancy.
Well, I think it probably is, as you describe, it's the cumulative patient contact and the closeness of the contact, and possibly also and this is just an anecdotal impression, but nurses are often having to restrain patients or move patients, or it's not like when the doctor arrives and stands discreetly, away from the bedside, out of airflow or coughing or yelling and screaming. Nurses are in the thick of it, and I one of my roles was to ring most of the positive cases of healthcare workers with the news that their test was positive. And I can remember very clearly, a nurse saying, look, I was wearing all the PPE, I was wearing a face shield, but I had to bend down and I turned my face, and the patient coughed just as I turned my face, and I was side on. So I think the lesson learned is the closer you are and the more contact you have, the more risk there is, even with PPE.
Yeah. And the other thing that really struck me was, the issue that was really hit the media last year around the tea rooms and their potential for COVID infections. And, you were talking about breakdown in infection control in doffing stations, but also in tea rooms. What were your thoughts around that? And I would have thought maybe more doctors would get infections potentially in the tea rooms, but maybe not.
Yeah, I think, again, I think some of this comes out when you've got a lot of virus around. So, under normal circumstances, the doffing and then sitting in the tea room, is fine if there's only one or two positive cases. But if there's an enormous amount of virus, because of patient load, it then turns up in places you don't expect it. I'd be interested in other people's views, but we formulated these ideas, in a very unusual setting, and that was a lot of virus. Yeah,
absolutely. Deborah, I might just go to you. There's a question for you from Catherine Keel. She asks, do you feel there's a role of antigen testing for travellers? There's often difficulty at present getting PCR results back due to time constraints for travellers, i.e. 72 hours within arrival at destination.
Yeah, look, that's a great question, and it's one that has, I know, been discussed at a number of different fora, and this is a woolly answer, I'm afraid, but it's yes and no. And so I think that there's a role if somebody is coming, from an area with very high disease prevalence, as I mentioned, but if somebody is coming into Australia, then we really want the most sensitive test that we can. And I have to say that an antigen test is not that test.
Yeah.
And we are fortunate here in that we do have good laboratory capacity for PCR testing.
Yeah.
And so, why would you not put your best foot forward, I think.
Absolutely.
And that's my own
Despite
opinion.
the delay in the result.
Well, I would say rather than use a test that has less sensitivity, why not look at ways of trying to reduce that time?
Yeah, and increase that. I had another question for you, which was really around saliva testing, particularly for children. So we know that at the moment around 50% or less of symptomatic children with respiratory symptoms are actually presenting for testing through things like the RCH National Child Health Poll and other sort of surveys and polls. So saliva testing in children is hugely appealing. Has there been much work done around that?
Yeah, I know that there has been a study at RCH, which has been led by Catherine Gibney and colleagues out there. I haven't seen the final results of that yet, but I can see that they certainly would have an appeal. But I can only comment on the work that we've done and others overseas, which has largely been in adults. I have to say that I would struggle to get saliva out of my toddler. Teenager, yes. Toddler, no. But
using a cheek swab, you could
using
get
a cheek swab, using a buccal swab, yeah, could certainly be useful.
There's another question for you, Deborah, from Phillips. Is rapid PCR viable as an alternative for travellers instead, given the cost?
Rapid
PCR, do you think he means rapid antigen testing? I don't know. Maybe that was what was asked before. Not sure.
Rapid PCR.
PCR?
Rapid PCR testing. Well, GeneXpert, as I mentioned, there are really marked supply issues with the cartridges used for that. I would just say that some of the high throughput PCR platforms now, which have are being used in labs, the random access ones, there's, for example, one called the Abbott Alinity, there's another called the Hologic Panther, and the turnaround times for those are less than 4 hours. And so we've really moved away from early January, where it was batch PCR testing, in-house testing, can take up to 8 hours, for a result to be put out. We've really cut that down. Yes, absolutely. Rapid PCR testing would be ideal for travellers.
Yeah, fantastic. Paul, it's great to still have you here. I thought maybe you had to dash off to another meeting, but there was a question I didn't get to ask you earlier, which Sonia Elliott had asked around the potential need for providers, immunisation providers or immunisers to have ongoing training in terms of delivery of the vaccines and the use of multi-dose vials, the APNA training course with the 6 modules. Do you think it's going to be enough for people to do the online course? I understand it's 3 to 4 hours just once, or do you think there'll be a requirement as we go through this rollout now to have ongoing training or sort of re-qualification for immunisers?
Well, I think it depends on your role. Really, the fact of administering the vaccine is pretty straightforward and just like any other vaccine that we do. So it's about the multi-dose vials I see as the real difference, and the handling when you thaw it and how you handle it before you thaw it that are the real new things to learn. So while I'm all for continuous education, and I think we should build these things in reading her question, I wondered if she's talking about longer term, so we're not always having to reinvent the wheel with every pandemic or something. I can see a benefit to that, but I do think we'll be able to get people adequately trained to do mass vaccination clinics and other settings pretty quickly.
Yeah. All right. So maybe just as we wrap up, Lou, maybe just lastly to you, any last pearls or thoughts you'd like to leave us with regards to healthcare workers?
So we're absolutely dependent on them, an obvious statement. And I think our experience shows that if you're hit by a wave with a high peak, things can go wrong. Whereas if you've got a flattened curve, a well-trained healthcare worker workforce can cope. But I think there are lots of things about transmission that change when there are a lot of infected patients in open areas, and the potential for aerosol as well as droplet transmission increases. And so I think as healthcare workers, we need to thank the public for all the sacrifices they've made, to flatten the curve, because for people in hospitals, it's terrifying when a large number of patients arrive all at once, and a whole lot of bad things can happen. Absolutely.
And Deb, maybe a last comment from you. There is another question for you on the capacity for genomic testing, all cases limits cut off.
So yeah, look, we're really fortunate here in Australia regarding the genomic testing, and I'm sorry I didn't cover that in the talk, but particularly here in Victoria, the MBU are sequencing at least 80% of all of the state's cases. What that does is give us a very granular picture of the transmission of this, and again, I never thought I'd see the day where a genomic sequencing report was being waved about by the Premier in a press conference, and so again, this is something that's played, a really pivotal part in the public health response. So I'm not quite sure what the question is, but if it's whether I think that all cases should be sequenced, yes. Limits, no limits. Yeah.
Okay, brilliant. All right. I'd particularly like to thank our speakers for this session, Paul Effler, Lou Irving and Deborah Williamson, and just to thank everyone for their time so far this afternoon.